Hello student, welcome to Anu's classes. Let's start today's video with another question paper of biology, and this is the question paper of 2017, and today I am going to discuss the botany section of the paper. So come to the first question, who discovered plant nucleus? And the answer is in 1831, the plant nucleus is discovered by Scottish botanist Robert Brown. Next question number 2, who discovered transpiration pool and cohesion of water theory? And answer is Dixon and Joly. Third question, what is the full form of EMP pathway? And the full form is Amden-Meyerhoff-Parnes pathway. Then question number 4, define the autotrophic and heterotrophic nutrition with example. And this question is carrying 2 marks. Then answer is, this is the answer, the autotrophic and heterotrophic nutrition. So, yeah. The autotrophic nutrition means what? In this type of nutrition, the organism prepare their food by own. Without means other help, they can prepare their food by themselves. And heterotrophic means in this type of nutrition, organism don't prepare their own food. Autotrophs are the producer in the food chain, whereas heterotrophs are the consumer in the food chain. Next, autotrophs use simple substance to form from the surrounding to produce food, whereas heterotroph consume the organic compound produced by the autotroph. An example of autotroph is plants, and whereas example of heterotroph is animals and another some plants also. Now go for the next question, that is question number 5. Draw and label the moss capsule. And so what is capsule? If question comes, then you have to write each sporophyte plant compose a capsule which is the center of spore formation. Means within the spore means capsule, in the sporophyte, the spore formation takes place. Next, this is the diagram of the capsule. So this is the diagram of moss capsule. Next, go for the next question, that is just a minute. Yeah. Mention the 4 major characteristics of prokaryotic cells. So you know prokaryotic cells means single cell structure, means unicellular structure. So first point is they lack a nuclear membrane. Since they have no definite nucleus, so no nuclear membrane is present. Second point is mitochondria, Golgi bodies, chloroplasts and lysosomes are absent. Third is the genetic material is present on a single chromosome. Next, the histone protein, like important constituent of eukaryotic chromosomes, are lacked in them. So these are the main 4 characteristics of prokaryotic cell. Next question, what is the specific rule of IAA? The full form of IAA is indole 3-acetic acid. It is a growth hormone. It is a type of auxin hormone. So IAA is the main auxin in plants, regulating the growth, development, cell process such as cell division and elongation, tissue differentiation, apical dominance, and response to light, gravity, and pathogen. So these are the main function of IAA. Sometimes question maybe asks what is the full form of IAA, then you have to write indole 3-acetic acid. Next, question number 8. Mention 2 examples of each of long day and short day plant. In my previous question paper, I have discussed what is long day plant and what is short day plant. So here I'm just asking the examples. So for short day plant, you have to write gentium and chrysanthemum, whereas for long day plant, you have to write radish and Spanish. Next, differentiate between dicotyledon and monocotyledonous plant with example. So this is the differentiation. So monocot means monocot embryo with single cotyledon. Monocot means embryo with single cotyledon, 1 cotyledon. That's why it is called monocot. Dicot means embryo with 2 cotyledon. Di means 2. So 2 cotyledon is present. Second, major leaf veins are parallel, means here the venation is parallel venation. Here is venation is of leaf is reticulate venation. Stem vascular bundle scattered, whereas stem vascular bundle is in a ring form. Root are adventitious root, whereas root develop from the radicle. Flower part in multiple of 3, flower parts in multiple of 4 or 5. Pollen with single pore, whereas pollen with 3 pores, and here secondary growth is absent, here secondary growth is present. So if 2 mark question come, you write 4 points, but sometimes this type of question will carry 3 marks also. That's why here I am giving you 7 points. Next, sorry. Then next, sorry. Next, go for the define inflorescence and explain the shift difference between the racemose and cymose example. So what is the definition of inflorescence? In a flowering plant, a cluster of flower on the branch or a system of branches is called inflorescence. Means the arrangement of flower, how they are arranged on the stem, that arrangement is called inflorescence. Inflorescence is of mainly two types, racemose and cymose. So first one, this one is racemose, whereas this one is cymose. So the difference is, so racemose, the main axis is ultimate growth, where the main axis is of limited growth. Flower arranged in a acropetal succession, flower arranged in the basipetal succession. Opening of flower in centripetal, whereas opening in flower is centrifugal. Usually the oldest flower at the base of the inflorescence axis, whereas usually the oldest part at the top of the inflorescence axis. It means, come to the diagram now again. So this is racemose. Here means flower grow from the base. Means suppose first in the base portion, this flower will bloom, then this flower will bloom, then this flower will bloom. So the older flower in the lower portion, whereas in racemose, first the stem will grow, and first at the terminal position, apical position, the flower will bloom, then the flower of the lower portion will bloom. So here the mature flower will at the apical portion, and in towards the basal position the younger flowers are arranged. But here the towards the here in the lower position the older flowers are there, and as the apical grow the younger flower bloom there. So this is the difference. Now next, go for the write the structure and the function of mitochondria with diagram. In previous question paper I have already given this answer, so you go through the previous question paper, then you get the answer. Then what is transpiration? Discuss the role of stomata in transpiration. Transpiration is a process that involves loss of water vapor through the stomata of plants. Since most of the water, 90%, is lost through stomata, plants regulate the degree of stomatal opening to reduce the water loss, provided that the plant has an adequate supply of next water. The water which is lost through the stomata is replaced by water from the xylem vessel in the leaf. During the day, when the stomata are open, the transpiration will become the major driving force in the movement of water in the xylem. Next, question number 13, the plant tissue system, and write the different types of vascular bundle found in the angiospermic plant with example. So first, what is tissue system? The plant's shell from plant tissue system that support and protect a plant. There are three types of mainly tissue system: dermal tissue system, vascular tissue system, and ground tissue system. And sorry, what are the different type of vascular bundle? Already I have discussed in previous year question paper, so you go through the previous year paper, then you will get the answer. Define the photosynthesis, write the prime differentiating characters of cyclic and non-cyclic photophosphorylation. This question is carrying 1 plus 2 plus 2 is equal to 5. Total 5 number this question is carrying, 5 marks. Now go, this is the difference of cyclic and non-cyclic photophosphorylation. In cyclic, only photosystem 1 is involved, whereas in non-cyclic, photosystem 1 and 2 are involved. No photolysis of water takes place. Here photolysis of water takes place. Oxygen not evolved, oxygen evolved. Here NADPH plus H positive is not synthesized, whereas in non-cyclic it is synthesized, and electron cycle back to the reaction center, whereas in non-cyclic electron does not return to the reaction center. And if you need the brief description, then you go through my videos where I have already discussed about what is cyclic photophosphorylation and what is non-cyclic photophosphorylation. Next, go for the next question. Describe the internal and external factors affecting the photosynthesis. So factor affecting the photosynthesis are given below. First, go for the external factor. These are carbon dioxide. Increase in the concentration of carbon dioxide can enhance the rate of photosynthesis. Second, internal factor, intensity. External factor is the intensity and quality of light affect the rate of photosynthesis. Third point, photosynthesis affected by the temperature. Photosynthesis is sensitive to higher temperature, as the higher temperature causes the inactivity of enzyme. Next fact, external factor, aeration is necessary for photosynthesis. In small quantities, oxygen is required for the electron transport. Next, external factor, nitrogen is known to have an impact on the rate of photosynthesis. Next, air pollutant decreases the photosynthetic rate, and soil, pH, and other climatic conditions play a role too. There are some internal factors also which affect the photosynthesis. Some of these are the internal factors: leaf anatomy, chlorophyll, plant hormone, accumulation of end products, performance of Rubisco, enzyme activity of the Calvin cycle, rate of chloroplast electron transport system, transit of stomata, trade of stomata, and carbon utilization, and the capacity of metabolite transport process. So these are the internal factor, and these are the external factor which affect the rate of photosynthesis. Okay, students. So in my next video, I am going to discuss the zoology section of this paper. Thank you.